Hello, and welcome to Cyclica's AI Ate My Homework. If you're joining us for the first time, this is a 7-part webinar series that will be providing key perspectives in science, technology, entrepreneurship, and the exciting field of AI and drug discovery. If you missed our first session last week, a link to the recording will be shared with you via email. Be sure to follow Cyclica on LinkedIn and Twitter, where we'll be announcing new topics on a weekly basis. If you have questions during this webinar, please use the Q&A feature and questions will be answered at the end of the session if time permits. Today, it's a pleasure to introduce our panelists, Vijay Shahani and David Kuter, who will be discussing drug development fundamentals and computer-aided drug design.
Great, thanks. Hi, everyone. So my name is David Kuter. As Devesh mentioned, I am the Director of the Scientific Computing here at Cyclica. And I suppose my background is I have a PhD in chemistry from the University of Cape Town in South Africa, where I did a sort of tried to understand how an antimalarial drug interacts with this target using both computational and experimental techniques. I then went on to do some postdoc work, doing more computational work, simulating biosystems like membrane lipids, and a bit more data sciencey type work at my postdoc at McGill University, where I worked at synchrotrons and tried to look at how heavy metals accumulate in bone fragments. And so my background is very much in terms of biology and using computation, using computers to try and understand disease and to try and develop drugs that could combat disease. Vij?
Thanks, Dave. Sorry, I was just looking for the unmute button. So hello, everyone. My name is Vijay Shahani. I am the Director of Applied Science at Cyclica. I've been with Cyclica for around over 4.5 years now. My, background or academic background was in medicinal chemistry. I got my PhD from the University of Toronto. At that time, I was looking at the design of so-called small molecule inhibitors for a proto-oncoprotein called STAT3. So to break that down a little bit, that is, I was designing potential drug molecules to target a protein target that is linked to cancer. And by finding molecules that hit this molecule, we were hoping to come up with a new treatment for cancer. During my PhD, I discovered a passion for visualization of science, and that led me to my master's in biomedical communications, also from the University of Toronto, where I learned how to use traditional media such as carbon dust paintings, pen and ink illustrations, to using digital software like Adobe Photoshop Illustrator, and all the way to kind of 3D animation, the basics of 3D animation, interactive media design, UI, UX. And it's really the combination of these two disciplines that kind of led me to Cyclica, where I've had a chance to apply both the application of the technology and also help build up the technology, which has been a wonderful journey for me. So kind of moving into the core of the talk today, I want to start off by doing a quick recap of some things that we learned from the previous lesson from Shelley and Ben. Shelley spoke about kind of the biology of proteins and disease. Just a reminder, proteins are kind of the functional unit of the body, so-called the body's workhorses. And it's through proteins that primarily most drugs work. And so, looking at this quick diagram, drugs may interact with a given protein. This will then have an impact on kind of cell behavior in some cases. That's if it's a anti-cancer drug, it might be something that kills cancer cells specifically. This will then have an impact on the organ or tissue level, and then it has an overall impact on the health of the organism on the body level. The other recap was the topic that Ben covered, which is kind of the fundamentals of chemistry from the sense of what chemistry do you need to know in order to find drugs that bind proteins. And so fundamentally, the same rules that govern kind of the interaction between a small molecule and another small molecule, or a protein and a protein, are the same. It's these intermolecular forces that hold proteins together and allow small molecules to bind. And he went over kind of three major types of intermolecular forces: the hydrogen bond, which is highlighted here in green. We have the ionic salt bridge, which is highlighted in blue. And then something called the van der Waals, or London dispersion forces is another name for them, where you have kind of this interaction between kind of non-polar groups. Just want to quickly kind of pull out some content from Ben's slides. So he kind of visualized this. In orange is the small molecule drug. In white, these ribbons are alpha helices that are the protein. And he highlighted in green here the hydrogen bond that's kind of leading to this interaction, and in blue, the salt bridge that's also important for this interaction. So these are two kind of topics that are kind of fundamental to kind of drug discovery, and we'll come back to this a little bit later in my presentation. But the real question is, how do we, discover drugs today? And so what I wanted to do was first kind of give a little bit of a historic perspective, and really what we can do is borrow as much information from nature as we can, so learn from nature. In fact, organisms have been evolving small molecules that modulate proteins for millions of years, right? So, things like, ATP even, or signaling molecules that Shelley touched on last week. So, there are an abundance of small molecules that can elicit biological function. And I just wanted to kind of show a couple examples here of what I'm talking about. So these three molecules on the screen, they may look a little bit complex and scary, but essentially what we're looking at here are three molecules from plants. And these molecules are used to kind of defend the plant in some fashion, so against a herbivore. Obviously, a plant can't run away, and so it has to defend itself chemically. And so what we have here is psilocin hungdushan, which is from a plant, Himalayan yew, and digitoxin. And each of these kind of molecules elicit a response in another organism, right? So the herbivore that's eating that particular plant. Interestingly, each of these naturally occurring molecules or phytochemicals are actually the basis for drugs that we have used historically and even today. The one on the far left is the basis of acetylsalicylic acid, which is aspirin. Paclitaxel, which is a taxane-based drug, is used as a chemotherapy agent. And digitoxin is, a cardiotonic steroid that actually is being used as a cardiovascular drug. And you'll notice if I flip back to the previous slide, flip back to the previous slide, that some of these look a little bit different, meaning that we went in as humans and modified these chemicals. And one of them looks identical to the source material, which is digitoxin. And that's just speaking a little bit about the drug discovery. So I guess the source of drugs today can be inspired from nature. So I wanted to go over this example because I always think it's a pretty interesting example of the discovery of a very important molecule, in this case, penicillin. And so Alexander Fleming was a very observant scientist, and he actually found an interesting observation in an experiment that he's running. So on the right here is a graphic of a petri dish that is covered with small colonies of bacteria. That's the yellow dots. And what we see here with these brown dots are these little mold spots. And Alexander Fleming noticed that around these brown spots was this halo where bacteria was absent. And that might have been something that was overlooked or maybe just dismissed outright, but it's actually a pretty curious result because the mold wasn't there. So why was the bacteria dying? And so upon closer investigation, Alexander Fleming was able to isolate penicillin, which was essentially one of the first antibiotics that we've used kind of on a global scale and actually revolutionized medicine. I think we kind of underappreciate the danger of infection and how deadly it can be, and that's mainly due or in thanks due to the abundance of antibiotics, where penicillin was kind of the progenitor of a lot of the antibiotics we have today. And so if it was this kind of, I guess it seems pretty easy, you just find a molecule that has a certain effect and then you could just have a drug. Obviously, it's not that simple. There's a lot that goes into the discovery of these molecules that could turn into drugs. So maybe let's take a little bit closer look as to what really we need in order to kind of start an investigation for discovery of drugs. And really at its core, there are two major things that you need to start a drug discovery program. And this is really simplifying things, but you need some source of chemical matter, right? So you need some type of molecule or collection of molecules to test. And then you also need a way to test them. So some kind of model system or some type of understanding of the disease. So let's look at the chemical matter. And, historically, early drugs, and you could even think of, food as a type of drug, an example being, people who were subject to long voyages on ships would often succumb to scurvy. And it turns out scurvy is just a lack of nutrients, in this case, vitamin C. And the treatment for scurvy was citrus fruit, particularly oranges, right? So foods in some sense can be, a drug. And another good example is people who are anemic would eat really iron-rich foods to supplement their diet. Getting a little bit more sophisticated are extracts from plants. So this could be, things like that willow bark that I mentioned earlier, which is the source of aspirin. Or even today, we're lots of research is being done in the phytochemicals or the plant materials that's found in the Amazon, or sources like jellyfish that are unique and have their own type of interesting molecules that could have or elicit a biological effect. Really kind of around the time of kind of the industrial, when industry really started to develop new molecules that were not organically based, was actually driven by the synthetic dye industry. And so this is when we started to actually, as humans, start to make molecules for very specific purposes. And from that understanding of being able to make molecules, we started to develop large collections of chemical matter that can be the source for new drugs potentially. So these are man-made compounds. And just really quickly, when we talk about testing frameworks, what kind of testing frameworks do we have, right? So one testing framework would be, either eating things or applying these extracts to the body and seeing what happens, right? So this is kind of at the organism level. That's, risky, right? So if you happen to ingest the wrong thing, that could actually be lethal, right? So you could, cardiotonic steroids is an example of something that you don't want to ingest in large quantities because it's going to stop your heart and you'll ultimately die, right? So we've gotten smarter. So this is kind of like the primitive level of testing. Eventually, we do need to get to the organism level at some point, which I'll touch on later. But at the early stages, we like to use models or proxies for discovering medicines. So there are these things called tissue or cell-based tests, so-called phenotypic screens or biological assays. And I'm going to talk a little bit more about that in the next step. And then, as we've kind of alluded to in the first slide and from Ben's talk and Shelley's talk, proteins are fundamental drivers of biology. And if we could have an understanding of what proteins are driving disease, we could actually target these proteins directly. And so this is kind of more of a knowledge or rational-based drug discovery program where we're looking at either proteins in isolation and having it at the bench in a wet lab, or we could do it in a digital fashion using, computer-aided drug design, which is something that Dave will talk about a little bit later. And so these are kind of different ways to kind of start seeding the drug discovery process. So what did I mean when I said cell-based models or phenotypic screening? So a large component of drug discovery happens before we even start looking at chemical matter. It starts when we actually are learning about disease. And this is why an understanding of biology and a deeper understanding of biology has made us so much better as a field at finding new medicines and helping people. And so by understanding fundamentals of disease, we create kind of these models or proxies for biology. And this allows us to test molecules at scale. If you're only going to be able to test one molecule at a time, a bit of drug discovery is a numbers game. You're going to maximize your chances of discovering something by being able to test multiple compounds at once. And so in this case, that this figure here, we call this a multi-well plate, where you have multiple small wells that contain a specific kind of biological test. And by applying different molecules to each of these wells, we could start to detect things that are active or inactive. So to go into a little bit more detail in that regard, let's say this is a cell-based assay whereby we're looking at cells on the left. These are healthy cells, maybe they're cancer cells perhaps, that are surviving and proliferating. To this well, we would add a collection of small molecules, right? In an inactive molecule, nothing would happen. Those cells would stay the same. There would be no readout. And that's the kind of end of that or that would be kind of a dud molecule or an inactive molecule. But perhaps you add a molecule and all of a sudden it causes a biological effect. And let's say that you cause these cells to start to die or cause necrosis. And this will have some type of readout, right? So in this case, these dying cells, maybe it releases some type of dye or some type of detectable output, which you can read on the plate. So, here in magenta is kind of this inactive well. There's the molecule doesn't change the behavior in any way. And in this green well, we have a molecule that elicited this, cytotoxicity, and we're able to detect that within the assay, right? And so you would do this, I'm showing here, how many ever wells there are, but in some efforts, in high throughput screening efforts, this could be testing thousands of compounds, if not potentially millions. Thousands more likely, millions is more stuff that we do in a digital format. Okay, so that's a little bit more about phenotypic screening and how we might discover what we call hit molecules. So this is way before something becomes a drug. So what's the next step? We found a hit, we found something that shows some type of activity that is desirable for a therapeutic molecule. So just like kind of, as I mentioned, organisms have evolved molecules over time. Scientists need to now evolve this molecule over time to make sure that it has the right kind of behavior. And so what are the things that we have to consider, right? One of the things, the first steps is to confirm that this molecule is actually active, right? Maybe it was a false positive. And oftentimes we will do this by testing the molecule in what we call an orthogonal assay or some type of counter screen, where we would then elicit and make sure that the molecule has the activity that we want. Okay, so that's one quick step, and that's usually pretty straightforward. The other things that we need to confirm is that this molecule interacts with the body in a favorable way, right? And I'll get into that in a little bit more detail in the next slide. And then the last question is, in the case of a cell-based or phenotypic assay, we don't have any information as to what is that underlying protein that is eliciting the response that I'm seeing. And oftentimes that's a very big piece of information or key piece of information for us to actually improve upon the drug's activity. Okay, so, what are some key kind of considerations that we now need to think about when we're kind of evolving this hit molecule, right? And sometimes this is maybe a little bit later on, maybe when we have a lead molecule, but it's still a major thing to consider. And the earlier we could start considering these elements, the more efficient we can be in discovering medicines. And so, what are the things that you want to consider? So is this, molecule, is this something that I could take orally, right? Can I ingest it? Or do I have to have some alternative form of application? Maybe it's something that needs to be done intravenously. Another question is, if I do ingest this molecule, is it readily absorbed in, let's say, the stomach or the small intestine, so that that molecule actually is bioavailable and gets into my system? Another question is, if I do get this molecule in my body, how stable is it going to be? Is it going to be metabolized by my body's, defense systems, or, the C4, the cytochrome P450 enzymes, which are kind of the body's way of breaking down xenobiotics, which are, any chemical that's not native to the body? Will this, system quickly eliminate these drugs and therefore make them completely inactive within my system? Or maybe it's the other way around. Maybe I need to metabolize this drug in order for it to be active. Another question is distribution. Once I absorb this drug, is it going to be carried around the body and arrive in the compartment that I want it to be? Perhaps it's something that needs to get into the brain. Is it going to pass by the blood-brain barrier, which is a big barrier, as the name suggests, to getting drugs into the brain and allowing it to be active in conditions that are neurological, or things like glioblastomas, which is a brain cancer. Do the drugs cause a toxic effect? Does taking this, molecule cause, necrosis or some type of renal dysfunction, right? And maybe that's mediated through interactions with things that were not anticipated at the early hit discovery phase of the program. And lastly, is this something that the body can eliminate? Will this molecule get removed from my body, or will it accumulate and potentially also cause a toxic effect? And so this is just kind of a, this is not exhaustive by any means. There's a lot of things that go into this, but these are some considerations. And there's an acronym here that's kind of hidden. There is ADME, which is ADME. It's one that is kind of common within the drug discovery space. Toxicity, or sometimes called ADME-T or ADMET. And these are different kind of principles that help guide the drug discovery process. And it's also linked to something called pharmacokinetics, which is a big word. We won't really get into it today, but it's all related to how does the body impact the drug. So what's the influence of the body on the drug itself? All right. And so what's the other major consideration that I mentioned, and kind of circling back to that early slide? It's whether or not this molecule binds to a given protein target. So turning back to penicillin, we find that, penicillin's mode of action is on this particular protein. And by understanding kind of how penicillin interacts with this protein, scientists were then able to evolve a whole collection of antibiotic drugs, first starting with penicillin, and then systematically modifying these drugs over time so that they are, better at binding this particular protein. And, a major kind of philosophy for a long time within the drug discovery space is make your drug as sticky for the protein of interest as possible. In that way, you can minimize side effects. Now that philosophy is changing slightly, but it's still an important guiding principle for a lot of drug discovery programs, is to try and maximize the chance of you binding the desirable on-target protein. And this is something that Dave will talk in a little bit more detail in just a bit. So to summarize, we talked about, the discovery of hit molecules and how we could look towards chemical matter and testing them in particular frameworks to discover hits. And then we talked a little bit about the considerations that you need to have to take a hit and convert it into a drug. And so that's, again, how does the drug interact with the body? Both how the body influences the drug, as well as how does the drug influence the body. It's a two-way street, and both are important considerations. The other kind of major concept is how does that drug impact that protein? What's the driving intermolecular forces that's leading to that interaction, and how can I maybe optimize this or improve this so that I get to a better drug molecule? And oftentimes these two things are kind of at odds to one another. It's a very challenging thing to optimize one without sacrificing another. So that's why oftentimes taking something from a hit to a drug requires multiple iterations and sometimes thousands of molecules from a hit to a drug. Again, evolving that molecule from a hit to a drug. It's not as simple as we found a hit, let's run with it. Usually it takes multiple generations and multiple iterations to get to that molecule, which is something that I know Dave is going to touch on as well. And the last thing I will leave you with is, we talked about the importance of maximizing, the ability of a drug to hit a particular protein. But again, the body consists of thousands of proteins, over 20,000, roughly 20,000 proteins that are in the body. And a drug molecule that you ingest has the potential for interacting with all of these things. And so another kind of notion is, how does that drug interact with other proteins within the body? Is this something that we could discover and use to better make medicines moving forward? And so with that, I think that's the topics that I want to cover. I will now pass it over to Dave, and thank you for listening to my portion of the talk, and I look forward to some questions later on in the session.
Yes, so as Vijay said, I am going to be talking a little bit about how we use computational methodology to influence the drug design process. But before I get to that, I thought let's maybe take a step back and address topics that are possibly more traditional, although I put this in inverted commas because the use of computers and the use of computational technology within drug discovery is very widespread. It's been used since the 1970s. It would be very hard pressed to find something or a drug discovery program that doesn't use some sort of computational technology in the drug discovery pipeline. And so what I'm really going to be talking about is a non-computational based method that has routinely been, that is sort of underpins a lot of medicinal chemistry. And this is the concept which is known as structure-activity relationships, where you take a hit, let's say, as Vijay mentioned, and you systematically change the molecule and assess how that change to the molecular structure impacts its activity. So for example, let's take a hypothetical molecule that consists of 3 green circles. You test it and you get a value that comes out of an assay, or you test its interaction with a protein, and you know that this molecule interacts or is a hit. But then you go on to say, well, let's try, can we get something better, or can we get something that is more or less toxic, or will have better ADMET properties, etc. And to know this, what you really do need to know is where on the molecule you can modify such that you can move towards these better properties. But also what you want to know is what parts of the molecule really are important for maintaining that activity. And so you go through the stepwise process of changing certain aspects of a molecule. So for example, in this hypothetical case, we can change one of the green circles to a blue square. And then we would take this molecule, you would synthesize it, you would test it, and then let's say in this case, you find that actually you have better activity. So it's doing even better than your hit did. But now you don't really know, is it because you have a blue square present simply in the molecule itself or at a particular position? And so what you then do is perhaps go and say, well, let's take a blue square and instead put it at position number 2, in the middle position. And what you find here is that actually it gets slightly worse. So it seems to be from these, from these very few tests, you're starting to get some sort of information about how changing the structure affects its activity. For example, what happens if you have two blue squares. And that, in this case, seems to give you activity that's similar to the starting compound, but you know, compared to this second molecule you tested, that you've actually decreased the activity. So having a square in this position actually is detrimental to its activity. But what about, let's go one step further. What happens if we get rid of the green circles altogether and we have a molecule that's entirely blue squares? And then you'll find all of a sudden something interesting will happen where you will break your activity. You will no longer have a molecule that will interact strongly or be active. And so this is actually very important because it tells you at what point you need to stop and what points and what things are very important. And in this case, it seems like having a green circle is important. But once again, is it just having a green circle anywhere in the molecule, or is it having a green circle at a particular position? So you would go back and you'd synthesize a molecule, something like this, where you'd put the green circle in the middle, test it, and you'd find, in this case, it still has no activity, which tells you that having a green circle in this position number 1 is very important for activity. And of course, you can go through various iterations of this. There are various different shapes and various different colors you can try. So you would often go not only just to blue squares, maybe what happens if you try a red triangle? In this case, it tells you that the red triangle is bad, but again, is it bad because it's a red triangle, or is it bad because it's in that position? So you'd go and you would make another one. And in this case, you would see that it's okay to have a red triangle, provided that you have the red triangle in the middle position, and that's actually really good activity. So based on this sort of fictitious molecular system, what you could then do is, based on this very, very small data set, is propose a new molecule and say, well, if we know we have to have a green circle at position 1, and we know that a blue square is good at position 3, and a red triangle is good at position 2, perhaps this molecule here is going to be really, really good. And if you make that, test it, and if you're really, really lucky, you'll hit the jackpot and this will work. Of course, this is all, this is very hypothetical, and what I thought would be perhaps a bit more interesting is to show you a real life case of where this has occurred. And this has occurred many, many times in just about every drug discovery company that you can come across in it, and most academic labs that are looking into just drug development. So I'm just going to give you an example that I know pretty well, which is of chloroquine. You may be familiar with chloroquine. It was recently touted as a possible drug that could be used to combat COVID. But it's probably most well known for its antimalarial activity, and this is the compound that I did my PhD on. But while it was known to be a very, very good antimalarial drug, why it was a very good antimalarial drug was not really well understood in terms of its structure. And so a group decided to do this sort of structure-activity relationship approach to determine what parts are good and what parts are bad on the molecule. So what parts are responsible for activity, and what parts could be changed to make maybe another molecule with similar activity or better activity, or in this case, make a molecule that could overcome resistance in parasites, because parasites had become resistant to chloroquine. So what the researchers had done is they'd taken this molecule and they tried to break it down into something a little bit smaller. They essentially just made this chain shorter and simpler, so made it 2 carbons and a nitrogen as opposed to 3 carbons and a nitrogen with 2 carbons on either side. And then they tested it, and what they found was that even though they truncated this side chain, this molecule still retained antimalarial activity, or at least activity on the target in the lab. What they then did was to say, okay, well, if we truncate it, let's truncate it a little bit more. And so they took the molecule and got rid of the side chain altogether, left the nitrogen on there, and they found still it is this molecule is active. This very, very small and seemingly simple molecule is active. So what they took it one step further. Okay, what happens if we remove, for instance, this chlorine? And what they found is as soon as you move that chlorine, you've killed it. It no longer has any antimalarial activity. It does nothing. And so it seems like this chlorine is very, very important for antimalarial activity. So one of the questions that they asked was, well, is it the chlorine that's really, really important, or can it be anything? So they then went back to this sort of first molecular fragment, and they decided to change the position of the chlorine. So change it, in this case, to a nitro group or a CF3 group, which retained activity. But when they changed the nitro to an amine or a methyl group, they found they didn't have activity. And so they could build up this picture of what was important and what wasn't important. And based on the properties of the types of things that they changed, they found out that having certain types of groups could give rise to beneficial antimalarial activity, while other types of groups could not be done. And in fact, what they found, what we do know is that if you change the side chain of chloroquine sufficiently enough to look different, you can overcome malaria resistance to chloroquine, because it no longer looks like chloroquine, but it still acts like chloroquine because it has this moiety over here. So that's just an example of how one use of structure-activity relationships could be used. And of course, as I mentioned, there's plenty of others that are in line with that. The big problem with the structure, so as powerful as the structure-activity relationship type methodology is in drug design, the problem with it is that it tends to be rather slow and labor-intensive. So you need to make all of these drugs and then test them in a system, and this becomes very expensive. As well as slow, you can't really access very many molecules. And so ideally, what you really want to do is to try and increase the efficiency of this system. And this is really where computational or computer-aided drug design came into play, where they were trying to take the burden off the lab chemists to create these molecules and test them to see whether they would be active or not. By making them virtually, evaluating them virtually, and then saying to the chemists, okay, well, we've evaluated a lot more compounds than you could. Here are our top predictions. Take these top predictions and go and synthesize them and test them. And hopefully get something that, maybe would have taken a couple of years to do. In this case, it took a number of years to do this sort of thing. Maybe you can do that in a couple of months. And so that drastically reduces the amount of time, effort, and energy, and of course, increases the efficiency and productivity of getting a drug from a concept, or getting a drug from concept to the market. So let's talk a little bit about how computers have helped this process. And probably one of the most well-known types of computer-aided drug discovery methodology that is used in the industry is that of what is known as docking. So docking is essentially when you take a protein that you're interested, that you want to target, and this is an example of a protein that is been rendered inside a computer, and we can describe this protein using various physics equations. And you take the protein, and then you take a molecule and you place it inside the protein or at a particular position in the protein, and using this methodology, figure out how well it interacts with that protein, how well it fits, how strongly it's predicted to bind. And then you do this for a large number of molecules, right? More so than we can do in the lab. And so, for example, an example of this is something known as virtual screening, where you can take a library of known compounds that are commercially available, that you can just buy off the shelf, and you put these into the computer, you run docking on all of these molecules, and then you rank them in terms of the worst to the best, in terms of their docking score. And then you can, once again, take these shortlisted candidates and move with them to see if they could be possibly hits, or they could be drugs for your particular disease. And this is really, really useful, particularly for something that is known as drug repurposing, where you're taking existing or known drugs, compounds that are used for another purpose, and repurpose them for a different disease. So a good example of this is COVID-19. Obviously, what we want to do is we want to try find if there are any known drugs out there in the market that can be used to fight this disease, for example, like chloroquine. And the reason why this is so attractive is because if this works, if the molecules are very active, you can reduce your time to market, because all the work has already been done in terms of safety testing, of scalability and manufacturing. for this other disease. And so you can just take it and say, well, we want to use it not only for that disease it was originally designed for, but for this additional disease as well. Because the time it takes to get a compound from concept to the market can be anywhere between 7 and 10 years with a very, very high failure rate. And so if you can get over all of these steps, which causes a molecule to fail in its path to becoming a drug, you're more likely to get something very quickly to market. And obviously for COVID, this is very important because we don't have anything to address this disease. The problem comes in with virtual screening and docking is that what happens if you find so you screened all of these molecules, you've purchased them, you've tested them, and you find that they're active, but they're not great. They're only moderately active. They're not great. And this is a problem because if they're not great, they need optimization. And if they need optimization, you're probably going to have to go through structure-activity relationships. And so virtual screening can be a good starting point for structure-activity relationships on new molecules, but really it wouldn't really help in that respect. And so there is an additional type of methodology that can be used, which is called de novo design. And this is where you take a molecule that has weak or moderate activity, and you systematically go through and change the molecule like you would do at the bench, but you do this virtually. So for example, let's say we've gotten this molecule here. It is predicted to be weakly interacting with a protein. But then what you want to do is you want to augment it, change it. So you would then say, okay, well, what happens if we add another circle? What happens if we add a bigger circle? What happens if we add a circle and a square, for example? Make these inside, basically generate these in the computer, predict whether it'll strongly interact or not. And then usually what you'll do is take something that looks like this one's got a strong prediction. Let's take that forward to the next day, in the next generation. And so we evolve, as Vijay was telling you, over time, a molecule towards something that will be a better candidate. So ideally at the end of this sort of multiple evolution iterative steps, you would get out a drug that, or at least a molecule that the computer predicts will interact very, very strongly with your protein of interest. Now, this is great because this is essentially virtual virtual SAR. It allows you to circumvent the manual synthesis and evaluation portion, and can also provide you with novel drugs because you're not looking at things that have already been made before. You're looking at something completely new that's been generated by the computer in silico. The problem with this type of approach is that chemical space is so large that it becomes very, very computationally expensive to do this type of approach. But even so, you can still evaluate much more than you could have in structure-activity relationships, so it does have its benefits. So let's take a look and see what those benefits are. So it is somewhat faster than traditional structure-activity relationships. It is somewhat accurate. It will never be as accurate as the real ground truth of knowing whether something will or won't interact. There is always this toss-up between speed and accuracy in computers. So you want something that can be really, really fast so that you can explore a lot more chemicals, but you tend to do so at the risk of accuracy. And so with docking and computational approaches, you never expect to get a absolute hit, like the number one compound that you predict is definitely going to be a hit. It's a sort of an enrichment technique. You're sort of getting a higher, you're sort of trying to skim the cream off the top, so to speak. You're trying to get something that may or may not work. You're having a better probability of finding it, provided that you're looking at a larger chemical space. And through all of this, we've talked a lot about the docking, is targeting a single protein. And I'll talk, Vijay's mentioned a bit about this, and I'll talk a little bit more about this later. But artificial intelligence in drug discovery is also becoming a lot more popular these days. And that's because artificial intelligence is really, really good at telling the difference between two things. So for example, if you gave it a lot of pictures of a dog and a lot of pictures of a chameleon, it will be able to identify the features of each of these things, and it'll say, oh, for a dog, we know that a dog has ears, it has fur, it has paws. A chameleon has googly eyes, is green, and has no teeth, for example. And so if you gave it a picture that it had never seen before, it would look for these features and it would be able to tell you it's a dog or it's a chameleon. So in the same way, we can do this with molecules. We can take the molecules, we can say, here's a whole bunch that are active, here are a whole bunch of inactive. It looks for features of active molecules and features of inactive molecules. And so it can make predictions based on these. And I'm not really going to into too much more detail about this because we'll probably have a in-depth session on it in about 2 weeks' time or so. But what's really great about this is that it's a lot faster than docking. And so we can see a lot more, we can do a lot more with it. And it tends to be more accurate than docking is, at least in our experience, although this very much depends on the models that are, that are created. In terms of single protein targets, we talked about hitting one target at a time. The problem is, as Vijay mentioned, that a lot of your molecule that you're designing for one target doesn't necessarily only interact with that one target. It may interact with a whole bunch of other targets, and that may cause side effects. And so ideally what you should want to do is not only consider your target, but consider a whole bunch of other proteins that you may not want to target at all. So at Cyclica, what we do is we have a list of protein, or have a target we have in mind specifically that we want to design for, but we also have targets that we want to design against because we know they have bad effects, side effects. So let's just go ahead and summarize these techniques. In terms of structure-activity relationships, they're pretty inefficient, but they're highly accurate. And the problem with them is that you really can't look at very many targets or compounds. But when you go to docking, you can speed up this process, which allows you to look at sort of tens of thousands of compounds, although still relatively few targets at the cost of some accuracy. But with AI, what you can do is you can start evaluating millions of compounds for thousands of proteins at once, with relatively decent accuracy compared to docking. And so generally what you'll do is you'll move from one side to the other. You'll start off perhaps with AI, get a short list of compounds, move to docking, get a short list of compounds, and then give this to the bench lab chemists. So with that, I think I'm going to stop here. I think Vijay, you've got another slide, a summary slide. Vij, I think you're on mute.
Sorry, again, mute button. Yeah, I think it naturally just was kind of summarizing the talk in general. And so kind of the notion of what we covered today was because we were understanding medicines better today, or diseases better today, we understand the biology, we could come up with models and tests to evaluate many compounds, to discover hit molecules, to help drive the discovery of new medicines. Historically, we've relied on naturally sourced molecules, but more increasingly, or recently, or even in the last 50 years, I would say the majority of molecules are derived from our own man-made molecules. Although there is, a lot of work being done to still source natural molecules because of the diversity that we find within nature. And just because we could find hits from kind of these early drug discovery efforts, evaluating these molecules, maybe doing some SAR, this is just the tip of the iceberg, as kind of Dave's talk kind of pointed out. And a hit has to go through many cycles of optimization before it becomes a drug. Either through rational-based drug design or other methods to try and increase or improve the properties of that medicine. And I guess that last point is knowing the protein target of action. While it's not essential at the early stages, over time it becomes more useful to know what those targets are so that we can make better decisions and drive a rational-based drug discovery. So yeah, that's just kind of the quick summary. Anything that you would add, Dave?
No, that's good. I think that's all for me.
Great.
Fantastic. Then we'll move on to sort of our Q&A session. You guys have asked quite advanced questions, but I'm really excited to share these with our panelists. So I think we'll start off with one that's a little bit more career focused and sort of, more to the journey of our panelists. So the first question here is, what are some of the biggest challenges you faced working at Cyclica and working in an industry that's still in its early days and sort of the technology is constantly improving and changing?
Dave, you want to go first, or?
Um,
I mean, I could maybe start, and then if you want to add your own thoughts. So, you know, as you kind of mentioned in the question itself, it's still early days, and kind of the technology is constantly improving, which makes it really exciting. I think part of the challenge is knowing, kind of, knowing when the application of the technology is going to lead to success. So we're still kind of learning what are the best systems to apply this technology to, and there's a lot of experimentation that we do internally to kind of figure out, well, what are the targets that we feel are protein targets that we could apply the technology to? What are some of the questions that we could tackle with the technology? What is just kind of outside of the scope of what the technology could assist with? These are all kind of questions that, you know, we're exploring and kind of building up a better understanding of application. And that's, you know, essentially what my team does, is apply the technology. I think another big challenge is it's a black box to some degree, right? So when you have docking, as Dave was talking about, it's all based in kind of physical kind of rules, right? So it's actually evaluating things like force fields, which imply or inform you on, you know, hydrogen bond donors and energies of certain interactions. With AI, we don't have that benefit. It's a bit of a black box. Why is compound A good for protein B? Sometimes it's not very clear, which makes it kind of exciting because you're coming with novelty, and the AI is learning things that we as humans don't quite understand. But at the same time, it does make it a little bit difficult sometimes to make decisions. So that's another big challenge. Yeah, those are just some kind of thoughts. Dave, anything on your end?
I think from my perspective, I mean, coming from the sort of docking side, coming from a chemistry perspective, it's quite difficult, like Vijay says, to understand the application of what you can apply AI to, particularly AI, because that's not really in my wheelhouse. And so it's finding what is accurate, what is inaccurate, what are the limitations, what you can trust, and how do you evaluate all those metrics. And so we've got luckily we've got a really good team here that sort of focuses on that sort of perspective. So we can use their expertise quite a bit, but I'd say that's probably the biggest in terms of the industry. I'd say that's probably the biggest thing right now.
Great. For our next question, a little bit more sort of technically oriented. For sort of, I guess, computer-based approaches for drug discovery, do you need the crystal structure of your target protein, you know, to sort of anticipate what some of these predictions will be?
So I can only there are some tools out there that try to do this without the three-dimensional crystal structure. In general, if you've got a three-dimensional crystal structure, it helps. It's really helpful for us. We need some sort of three-dimensional structure. So whether if it's a crystal structure, or whether it's something that you've gotten from NMR, or something that's gotten or from cryo-EM, we need some sort of three-dimensional protein structure. We can use homology models, but the less certain you are of that protein structure, the less certain you can be of your results. Let's put it that way. But there are, like I said, there are some technologies out there that are trying to work around that by just looking at the protein sequence, say. But we're not necessarily too, at least in our experience, we find that having the three-dimensional structure gives better results.
Yeah, and I think one other quick thing about that is the notion that even X-ray crystals and these are snapshots of very dynamic systems, mm-hmm. right? So a protein, you know, you see these pretty pictures of these static crystal structures. That's not what a protein looks like in reality. In fact, it's rapidly kind of there's microscopic movement. There's things on the nanosecond scale. Those are very dynamic systems. And so unfortunately, with systems like docking, it can't possibly evaluate all these kind of permutations that the protein and the small molecule go through. And so while it can to some degree predict activity and can predict energies, it's not going to be perfect because it can't perfectly replicate the complexities of this dynamic system, right? And so in some ways, what we do at Cyclica is, our using AI, we've actually abstracted that a little bit and actually allow for more fuzzy understanding. So we're not as reliant on super high quality structures because we look at things more holistically. And I can't go into any more details, but that's one way that at Cyclica we're trying to accommodate or better kind of tackle these dynamic systems.
Awesome. For our next question, sort of touching upon developing molecules with, I guess, certain activity in mind, and whether that's done in a computer or cell-based assay. I guess, Vijay, you could talk a little bit about this, is, how do you sort of optimize for optimal pharmacokinetic properties and, making sure that, while you do have the desired activity with a protein, that, you're still developing the right drugs and, something that a human can take?
Yeah, that's a great question. So I think I did mention that sometimes, the optimization of a small molecule for a protein can run a bit at odds with, these other optimal ADMET properties. And so, in terms of kind of understanding pharmacy or big pharma has been, or researchers in general have been at, medicinal chemistry for a while. And so there are certain kind of things that have been learned over time. We understand better things like metabolism, properties of drugs that make them more orally bioavailable and things like that. And, adding functional groups in certain areas so that it's not as likely to be decomposed by your CYP proteins, et cetera. And so, there is, knowledge that you could install from kind of just the field and the many years that we've been doing medchem. And there are tools that we could use to actually help predict these features as well. So Cyclica has something called POEM, which is a predictive model based on, chemical structure and measured experimental data to predict, properties that are linked to ADMET, right? So is it likely going to be passing the blood-brain barrier, for example? And so balancing these two, both the affinity for target and ADMET is something that is important, and applying these two kind of predictive tools concurrently can often lead you to a better result. And so it in some ways, you may need to optimize a molecule, make it a weaker binder to the target, but then it's going to be a better drug overall. And that's very common, actually, is that the thing that ends up being a drug is not as strong affinity for the target.
Great response, Vijay. Really resonate with you on that. I guess our final question for this session, this is both to Vijay and David. What do you think the future scientists, and in terms of the tools that they'll be using in the future to do drug discovery, how do you see the relationship with AI, chemists, biologists sort of working together to develop the medicines of the future?
That's a really good question. So I mean, we can talk on sort of a overall level and talk on a more personal level as well. In terms of a more personal level, I suspect that there is going to be a lot more emphasis on programming coming into chemistry. So being able to analyze data, large numbers of data that are coming through various different formats. So if you can do more data science type work or creating programs to analyze your data, I imagine that that's probably going to start being a little bit more mainstream, as opposed to just running off to computational chemists to get certain things done. I think there's going to be a base there'll be a basal level of how to program, and I think that's always a good thing to know. Coming from someone that didn't know how to program just about at all in his PhD. But I also think that my whole sort of upbringing in terms of academia and education has been to have a multidisciplinary interaction. So people that are the people that are designing drugs are not just chemists. They're talking to biologists. They're talking to computational scientists. They're talking to clinicians. It has to be a holistic process in order to get from one stage to another, because it's becoming very difficult to find low-hanging fruit in terms of drugs these days. You have to go through and find more niche areas that are going to be, that haven't yet been tapped. So for me, it's always a case of, we're always going to need experimentalists to create drugs, to test them, to inform our models, but incorporating more AI, more computational analyses, and more biology into the whole process is definitely something I foresee. Peach?
Yeah, Dave, I'm not sure there's much I could add there. I think just kind of from a personal perspective, and I think people have to be open. A chemist should be open, and scientists need to be open to the tools that are being developed. Is essentially, I think it's more of a mindset thing. I think scientists are going to work hand in hand with these predictions to come up with better decisions, come up with more novelty, and ultimately, it's meant to augment and be used as a tool, just like another tool in the toolbox, like SAR was or docking was. I think AI becomes another fundamental tool that scientists are going to use in order to push the next generation of new medicines, right? In order for us to find and discover things faster and more efficiently. And that's just kind of a philosophy thing. I think that's, Dave got really practical with the skills and the like, and I'm just thinking more of philosophy. It's going to be something that we need to work together with.
Definitely. I think as a whole, Cyclica's philosophy is that AI is not going to be produce a silver bullet. It's not, it's going to be part of the process. It's going to have an important role in the process, but it's not going to be the thing that does everything.
Well said. I could not agree more. Thank you, Vijay and David, for sharing your insights on both your careers and expertise in drug discovery. Next week, we will have a special webinar with leading experts from renowned organizations who will be sharing their story and journey into the world of AI and drug discovery. Be sure to attend and engage with our esteemed panelists. It's an event you don't want to miss.